I'm the editor-in-chief of the MPMI journal, and this is our virtual seminar series that we've been having since the beginning of summer, in which we're highlighting different papers from the journal. We'll be continuing this. We have one more on December 1st. That's where I will actually be speaking, telling about our new, our search for the top 10 unanswered questions. What are the most important unanswered questions in our field? That's what I'll be talking about next time, December 1st. And in fact, we've written an editorial about that, which has just appeared today in our just published on the MPMI webpage, if you want to go take a look, preview. Then we'll be starting up again in January, and we'll be sending out some announcements for that. We'll have it posted so that you can register. I'd also like to introduce my co-host today, Kay Jones from Florida State University. Kay is a member of the MPMI editorial board, and she is also guest editor of our special focus issue that's coming up in which we'll be closing November 15th. So if you're interested, we'd love to have your submissions. The focus is on the #1 top 10 question that we identified, which is how do plants simultaneously interact with beneficial and pathogenic microbes? How do they do it? How do you let the good guys in and keep the bad guys out? How do you, plants can be pretty big. How do you manage that spatially and temporally? Really exciting, big open area of inquiry that people are diving into now. So we'd love to have your submissions, research articles, short communications, reviews, and Ashley has, I believe, typed the link in to the chat. So if you want to go and read more about the focus issue, you can just click on that link there. And yes, so Kay, do you want to take a few moments to introduce yourself, and then I will introduce our speaker for today.
Hi, I'm Catherine Jones at Florida State University. I'm on the symbiosis side of the equation, along with Jean, in terms of how plants discriminate between beneficial microbes and pathogens. And I focus more on the bacterial side, although I've done quite a bit with the interaction of symbionts with the plant, so, and pathogens. So thank you.
Thanks. And so this, represents some of the diversity of the editorial board. If you've come to many of these, you'll see that I have different editorial board members serve as co-host because we feature talks on different topics. So today's speaker, I'm delighted to introduce Sicard, who is going to be talking about work she did in Rodrigo Almeida's lab, and is now at Montpellier at the IRD. And she's going to be talking about a technical advance. And as you know, all the technical advances at MPMI are open access, so you can go read it, get all the details yourself once you have heard the talk, especially if you want to apply this. That's the whole joy of a technical advance, and that's why we made them all open access, because the whole point is that these feature new approaches, new techniques that people have developed, and we want to make that available to our members around the world, our colleagues around the world. And Ashley's typed the link to the paper into the chat, so you can always go there. But now I'm going to turn you over to Sicard, who's going to tell you about this very interesting technological advance focusing on the bacteria in plants. So thank you, Anne. Anne, you're muted.
Sorry about that. Thank you very much for the introduction. So I'm going to share my screen. Right. Can you see the screen?
Yes, it all looks good.
Perfect. So, hello everybody. So I'm going to present you one of the projects I've worked on during my postdoc in Rodrigo Almeida's lab, and which was developed in collaboration with Leonardo de la Fuente's group. So this project aims at determining a bacterial population growth status by using qPCR at a single time point. So the growth dynamics of a number of bacterial species has been extensively studied in vitro thanks to repeated measurements using either batch or continuous cultures. However, growth dynamics of bacteria in their natural environment is usually much harder to assess and is thus still poorly understood for a number of bacterial species. So the tools that we often use give us a snapshot of the colonization process, which is very important to understand how bacteria colonize their hosts, but do not inform on the bacterial growth rates. So one way to determine the bacterial growth dynamics during infection is to run time course assays. But such experiments can be time consuming, especially when working with slow-growing bacteria, and are not always possible to perform since it often involves disruptive sampling. Another way to address this question, which has recently been used by Corem and coworkers, is to take advantage of the relationship between bacterial replication and their growth. So indeed, most bacteria have a single circular chromosome, which replicates bidirectionally from a single fixed origin toward a single terminus. So this means that during DNA replication, regions that have already been passed by the replication fork will have a higher number of copies than the regions that have not yet been replicated. So in other words, during replication, if you're close to the origin of replication, you're going to have a higher copy number than if you're close to the terminus. So the ratio between the copy number near the replication origin and that near the terminus, which Corem and coworkers have called PTR for peak to trough ratio, should thus reflect the growth rate of the bacterial population. So for example, if we look at this figure, bacteria are in a stationary phase, which means that most of the cells are not growing. So their genome is not being replicated. And so we see that in this case, there is a uniform coverage across the genome, which means that on average, a similar number of reads are mapped across the genome. Whereas during exponential growth, the genome is being replicated, and so the number of copies at the origin of replication is higher than that at the terminus, as drawn here. And so this leads to a peak at the origin of replication and a trough in the terminus region. So two parameters are going to influence the peak to trough ratio you obtained. So as shown here, the higher the number of replication forks initiated, the higher peak to trough ratio you will obtain. And so since PTR is measured at the population level, the second parameter that is going to influence this ratio is the actual percentage of the population that is replicating. So indeed, especially in natural environments, bacteria can have asynchronous growth, and so only part of the population is actually growing. So as you can see here with the blue population, where only half of the population is dividing. And so the proportion of the population that is replicating and thus dividing is going to influence this ratio as shown here. So in order to determine whether a peak to trough ratio could actually be inferred from genomic coverage and to assess whether this ratio was correlated with bacterial growth rates, Corem and coworkers first carried out in vitro growth experiments on E. coli. So as shown here, they observed an uniform coverage across the genome during the lag and stationary phases, while during exponential growth, a single peak and a single trough were observed. So they then calculated the peak to trough ratios for each of these time points, which are here represented by this red line. So these peak to trough ratios were around 1 in the lag and in the stationary phases, while they reached 2.6 during the exponential phase, which indicates that there is multi-fork replication. So they further showed that the PTRs were correlated with the measured growth rate, represented here by this blue line. And they showed that PTRs preceded this growth rate by 30 minutes. So this indicates that PTRs predict the changes in abundance of E. coli. So the authors of this paper also confirmed this correlation between PTR and the measured growth rate for other bacterial species present within the gut microbiota. So based on this result, we decided to look whether this relationship was also verified for the slow-growing bacterial species that we studied in our lab, which is Xylella fastidiosa. Xylella fastidiosa is a gram-negative bacterium that causes significant damage on a number of plant species. So it is xylem restricted, and its only natural way of propagation is via insect vectors that fed on this tissue, which includes over 100 of insect species. Xylella is able to multiply both in its host plants and in its insect vectors. So this bacterium was first discovered on grapes at the end of the 19th century by Newton Pierce in California, where one of its main insect vectors is the blue-green sharpshooter. So this pathosystems has thus been studied for over a century in California and might be the best characterized Xylella pathosystem so far. However, as for most bacterial species, its in vivo growth dynamics is still poorly understood. So because of its fastidious nature, having a means to determine Xylella growth rates will be highly useful, not only to study its growth dynamics during infection of its hosts, but also to rapidly test the effect of various factors on its growth rates. Indeed, there is still no control method available once a plant is infected with this bacterium. As a result, we tested whether we could use PTRs to measure Xylella growth rates. So first, we ran in vitro experiments to look at the correlation between these two parameters, so between PTRs and the measured growth rates. So for that, we plated in parallel a Xylella suspension on several plates containing a rich medium adapted to Xylella growth. And so at different time points, we scraped all of the cells from one of the plates that will be suspended into 1 ml of buffer. And after a DNA extraction, we separated each sample into two. So one sample was sent for Illumina sequencing, while the second sample was used for qPCR. This experiment was repeated 3 times with 2 different strains of Xylella that had been previously isolated from grapes. So let's first look at the results obtained by NGS. So as previously, on the horizontal axis, you have the genomic position, and the vertical one corresponds to the genome coverage depth. And this is the growth curve we obtained from this experiment. So as you can see here, we observed a single peak near the origin of replication and a single trough near the terminus during the first 4 days of growth, while at day 6 and 7, when cells were in the stationary phase, we observed a quasi-uniform coverage across the genome. And we obtained similar results for the second strain of Xylella we tested. So while the pattern observed here is consistent with the growth curve, we found no correlation between the PTR values that were computed and the measured growth rates. And so because we were looking for a quick and inexpensive method that could be widely applied to assess the growth rate of a number of bacterial species, we decided to design a qPCR assay to measure PTRs. So for that, we designed 3 different primer pairs near the origin of replication of Xylella and 3 primer pairs near the predicted terminus. And we measure the PTRs obtained using this 9 combination of primer pairs, which are here represented by 9 different colors. So here we plotted the PTRs against the days. So for most of this combination, so for most of these couples of primer pairs, we observed higher peak to trough ratio during the exponential phase, which then decreased until reaching the stationary phase. So as you can see, we obtained PTRs above 4 at day 1 when using these couples of primer pairs, which could be an indication of multifork replication, but this is likely not to be the case for this slow-growing bacterium. And as you can notice here, PTR values around 1.6 were obtained during the stationary phase when using these same primer pairs. So this suggests that PTR values might not always be straightforward to interpret. So we repeated this experiment with another Xylella strain and observed a similar trend, although less pronounced in this case. So importantly, for most of the couples of primer pairs, the PTR obtained were significantly and positively correlated with the measured growth rates. And so the best correlations were obtained when using a lag phase of 1 day for both strains. So this suggests that PTRs precede and foresee Xylella growth rate by 1 day in these conditions. So what is important to return from here is that the PTR values obtained by qPCR are unable to measure Xylella growth rates. So since growth medium can affect bacterial growth, we then looked whether changing growth medium affects the correlations between PTRs and the growth rate. We thus ran a similar experiment using this time the PD2 liquid medium. So here you see the PTRs obtained for 2 of the primer pair combinations that robustly led to good correlation between PTRs and the measured growth rate, whatever the strain and the growth medium tested. So we obtained PTR values around 1.6 during the exponential phase and around 1 during the stationary phase. And again, a good correlation between PTRs and the measured growth rates was observed with a lag phase of 1 day. So changing the media did not affect the correlation between PTRs and the growth rates. So because all the ratios correspond to an average calculated at the population level, we then decided to look whether the presence of dead cells within the population could have an effect on the ratios. So we assumed that cells with damaged membranes should have a ratio of 1, thus lowering the average PTR during growth if present at a high proportion, while having no effect during stationary phase. So in order to measure the PTRs from only viable cells, we pre-treated the cells before DNA extraction with a reagent called Propidium monoazide or PMA. So this reagent is a DNA binding dye, which binds in a covalent manner to double-stranded DNA upon light exposure, thus preventing its amplification. So as this dye cannot enter cells which have intact membranes, it only binds to exposed DNA from cells which have damaged membranes or to extracellular DNA. So it thus should enable to amplify DNA only from viable cells. So as we have previously worked on a protocol to distinguish between Xylella viable cells from those with damaged membranes by using PMA, we used the conditions that we found were optimal in our previous study. So on this graph, you can see the PTR values we obtained after PMA treatments. And as you can see, this treatment had a huge effect on PTR values. It indeed both increases the ratios obtained during exponential phase, while decreasing the ratios during stationary phase. So while we do not have satisfactory explanations for the values we obtained after PMA treatments, we obtained consistent values across the 3 repetitions. Besides, these ratios were even better correlated with the measured growth rates than the one obtained using the non-treated samples. So there's higher correlation along with the increase of the PTR value range, ranging from 0.1 to 2.5 after PMA treatments, pushed us to PMA treated our samples coming from our in vivo experiments. Indeed, with a difference of at most 1.6, it can be hard to determine whether the cells are actively growing and what proportion of cells are growing, while it's much easier to determine this when a 10-fold difference is observed, as it is the case after PMA treatments. So to summarize, we have seen so far that PTRs can be used to predict the abundance changes of Xylella fastidiosa in vitro when using qPCR data. Moreover, we have shown that there was a better correlation between PTRs after PMA treatments and growth rates, and that the higher range of PTR values obtained with PMA pre-treated samples help better discriminate between growing and non-growing cells. So our next question was to determine whether PTRs could be used to assess Xylella growth rate during host colonization. So first, we mechanically inoculated grapes with a suspension of cells belonging to one of the 2 strains we had used for the in vitro experiments. And we sampled petioles at 3 different time points post-inoculation. And we pre-treated the cells with PMA before conducting DNA extraction. So as we could not measure the growth rate due to the disruptive sampling method, we plotted the viable cell population against the PTR values. So the first thing we can notice here is that the PTR range is similar to the one obtained in vitro after PMA treatments. So this suggests that the values we obtained in vitro were not due to particular experimental conditions and are similar to the ones found in natural conditions. Importantly, this suggests that the values obtained during our in vitro conditions can be used to interpret the values obtained here. So if you recall, we obtained PTRs around 0.1 when we had reached the stationary phase, and PTR slightly above 2 when most of the cells were actively growing. So apart from these samples, in which most of the cells might be dividing, in most of the petioles we harvested, we have PTR values in between. So we can assume that most of these samples contain a heterogeneous population with a proportion of cells that are actively growing and others that are not growing, as it might be expected during in-planta colonization. So these in-planta results suggest that by taking a sample at a given time point and 2 primer pairs, so one at the origin, one at the terminus, you can both determine the cell population, which is epidemiologically important in the case of Xylella, as it is positively correlated with its transmission rates, and determine the bacterial population growth status by using PTRs. So as Xylella also multiplies within the mouse part of its insect vectors, we conducted a similar experiment using blue-green sharpshooters. So we let the insects fed on a Xylella infected grape for a day, and then we transfer them to healthy basal plants that is not a host plant for the bacterial strain we used. So this was done to avoid potential reacquisition of cells by the vectors. And so we collected the insects at different time points after acquisition to measure the bacterial population they carried and the peak to trough ratio. So these sampling time points were chosen based on previous experiments and aimed at covering most of Xylella multiplication phase within the vector. So as during the in vitro and the in pronta experiments, we obtained a similar range of PTR values after PMA treatments. So if we look at these PTR values, we can see that in most of the insects, the ratios were comprised between 0.25 and 1.2, which suggests that as for the in pronta experiment, we have asynchronous growth of the bacterial population, with here a rather small proportion of the cells that are actively growing. So if we now look at the cell population, we notice that it is rather low even at 10 days after acquisition. So because blue-green sharpshooters can usually carry up to 10 to the 5th cells, we cannot exclude that the mature adults we use in this experiment were not in optimal physiological conditions, which may have impacted bacterial growth here. So to conclude, we have shown that while NGS showed the trends we expected to obtain during in vitro cross experiments, the PTR values computed were not correlated with the measured growth rates. On the contrary, our results suggest that by using qPCR, PTRs could be used to predict Xylella growth rates. Moreover, the results we obtained during plants and insect colonization suggest that PTR using qPCR data might be applied to determine Xylella growth status in its natural habitats. So we hope that this method will enable us to test the effect of different biotic and abiotic factors on Xylella growth dynamics. So while all these experiments were performed on a slow-growing bacterium, we believe that the method that we use might perform better on bacterial species that grow faster. And finally, although our method aims at determining bacterial growth rates at a population level, the correlation between replication and growth rates could be used to study growth rates at a single cell level, as recently shown by Hogan and co-authors by using fluorescent microscopy. So here the green dot corresponding to the origin of replication and the red one to the terminus. So with that, I would like to thank all the people involved in this projects, as well as the CDFA Pierce's disease program and the Marie Curie actions that funded my fellowship. And thank you for your attention.
Thank you, Anne, for a great talk. Just remember, everyone, type your questions into the Q&A. So very interesting to see this approach and to see how Anne used it for addressing the context, doing it in the context of a pathogen. Okay, so Catherine Jones and I will take turns reading the questions. I'll start. Juan Carlos Diaz-Vici says, nice and interesting talk, Anne. I wonder whether this technology can be used with fungal pathogens.
That's a great question. Unfortunately, I don't know much about fungus replication.
Yeah, it's so different, right? Because the whole advantage, although there are places where, you know, and eukaryotes where where replication starts and where it's late, but it's a lot more complex. Also the whole multiple chromosomes thing. Okay, so Kay, do you want to take the next one?
So the next question is, can digital PCR be of help to further improve this kind of experiment?
I guess it could have been applied. Whether it would have improved it, I have I have no idea, but
Well, oh, go ahead.
Okay, next question is from Dong Wang, and he says, very cool. Can this approach be applied to monitor the dynamics of a complex bacterial community, such as the soil microbiota? It's an interesting question.
well, we hope so. So the idea is basically to design primer pairs that are specific to each of the bacterial species. Well, if you use qPCR, but so if you can do so, yes, I think it could be applied for to look at the growth dynamics of microbial community. Actually, by using NGS, previous work, so Corem et al. And also Brown et al., a paper from 2016, showed that you could use this correlation between bacterial replication and growth rates to look at growth dynamics in microbial communities.
Well, I have a question. This was something that was so very interesting in the paper, that some of the peak to trough ratios that you got with the whole genome sequencing with the NGS. Well, you got a nice trough around the terminus for a copy number. There was a lot of variability in peak to trough across the genome, and it said that some of that correlated with areas of GC, with GC content, GC proportion. But I was wondering, so some of the ones that didn't correlate with GC content, did you have any predictions about structural variations in the chromosome or anything like that at some of those other sites that had atypical ratios?
Yeah, that's a really interesting question. Actually, we don't know. So the percentage of GC partially explain this variation of genome coverage, but yeah, there may be other explanation for it. Yeah, yes, could explain that.
So we have, I think this is a two-part question. From Caitlin Allen, I'm going to read the one from the chat, although we encourage everybody to write your questions into the Q&A. So Caitlin says, hi Anne, lovely talk. What would you expect to see in a vascular pathogenic bacterium with very different densities in a single stem slice? That is, would you get the same number from a stem with many partly colonized vessels as from another stem with half the vessels completely full? Yeah, that's an interesting question. Stationary, and the rest are just beginning to be colonized, early log. The complexity of tissues.
Right, that's why it would be like really interesting. I think it's a really cool technique for that, to actually, because we have a lot of heterogeneity during the plant colonization. So yes, if you might have heterogeneous cross across the sample, that will be reflected by the PTRs. I don't know if this answers the question.
So please, just to encourage people to please type your questions into the Q&A. Lots of questions, especially if you're thinking of applying this, as I think many people are. So what really caught me, and when I was listening to your talk, that I hadn't really appreciated when I read the paper, was the ability to look at bacterial growth in the insect vector, right? And this is how it mostly gets transmitted. And of course, one of the, it would be great to be able to treat plants that have been infected, but wouldn't it be better to be able to prevent infection, right? That's kind of a target. And so anything that so management practices that target the insect, this might really be cool. The whole point that you said that when you put it on basil, it may not have been that happy because it wasn't a host plant. You may not have had as much bacterial growth. You could try different ways of growing or treating the insect, and then use your method to see, to look at bacterial growth inside the insects. That would be a really interesting and very accurate way of seeing how likely, I guess, those insects are to be able to pass on the bacterium. Anyway, that was really more of a comment than a question, but I just thought that was really interesting possible application of this work that I hadn't appreciated.
Yeah, but you're right. Actually, there are several research programs that, so there are some that try to prevent or limit the infection within the plants, or the colonization of the plants, but you have also like a lot of work that is done to avoid either to avoid the transmission or to decrease the number of insects to decrease transmission.
Yeah, so instead of using disease as the assay output, you just test the bacteria and the insects directly. You know, what are the treatments that actually affect, you know, the ability of the insect to support the bacterium. So cool. Kay, do you have any more questions?
Well, I had a very practical question. I noticed from some of your previous work that where you'd looked at the effect of PMA on cell viability and found that under the conditions you used it, you know, it wasn't much unless you added enhancer and things like that. But I'm just wondering about sample processing time, and, you know, if someone's managing a lot of samples, that is it important to wait to add the PMA, you know, after you get into the lab at the time of isolation rather than doing anything in advance and things like that?
So the way we do it is to, so first we sample the insect or the plants, we grind them, and then we treat them with PMA, we pellet the cells and freeze them.
Okay.
So then you can actually move forward from that.
Mm-hmm. So do you want to read the next one, Kay?
Yeah. Do you see a way this new approach could help limiting the spread of this bacteria in the areas where it's threatening crops? And this is Lucas Bonametti.
So the idea of the technique is to actually look at the growth, to be able to quickly determine the growth rates of the bacterial population. So you could actually use this technique to assess the effects of different treatments, for example, on bacterial growth rate. So that would be a way to apply it.
Mm-hmm. So we have another question from Alba Pacheco. I have a very naive question. Do you think that the growth or replication state of the bacterium would change depending on the insect vector that you study? In other words, could using another vector species have a different pattern?
So it's possible that depending on the interactions between the bacterial strain and the insect species, you might have different growth rates. So it might impact the PTR values. So you might have, in some insect, it's possible that it actually grows faster in some insect species than in other ones.
It would be wonderful to see that dissected from insect organ to organ, you know, salivary gland and so.
Yeah, so Xylella actually infect only the mouth parts of the insect, so it's really restricted to the precibarium and the cibarium of the insects.
Interesting. Do you want to read the next one, Kay? From
Pascal Saldarelli. How can we you benefit from this technique to distinguish Xylella resistant from susceptible cultivars?
Well, so in resistant cultivar, you might have less growth, or actually, yeah, the growth might be, Xylella grow less in resistant cultivars, so you would expect to have lower PTR values than in susceptible. That would be my guess. And in susceptible cultivar where Xylella is going to grow more and better.
I just had a thought because, you know, I work on the Rhizobium legume symbiosis. And of course, one of the features in many plants of the infecting bacteria is that it stops being able to divide once it's mature, which is kind of an interesting little way that the host controls the bacterium. So it'd be interesting to use that to follow it. But I was also wondering, because then I realized Rhizobium does this endoreduplication. How would this method be able to detect endoreduplication? I'm not a microbiologist here, so I'm guessing that's similar to replication, except the bacteria don't separate. I don't know. I'm getting out of my depth here, Ashley, but okay, but that's what I'm thinking.
It seems like if it's complete, you know, because the DNA content measurements at with bacteroids versus the unreduplicated, those do assort usually into peaks. But I suppose if it was incomplete. So Anne, what do you think? With endoreduplication, you get multiple copies of the bacterial chromosome per cell. But I'm not sure that anyone has looked to see if that's always complete endoreduplication, and if the terminus is always amplified as much as the origin.
Interesting question.
So what do you think about that?
I actually don't know.
It'd be interesting to see whether you could apply this technique to endoreduplication.
Yeah.
No idea how that works. And I don't think anybody does, Anne. So many, I think one thing we're all seeing is lots and lots of applications of this technical advance to many different systems. Kay and I are already kind of mentally brainstorming how we could apply this to our own system. So apologize for that little thinking we're doing out loud. Dylan Beal has just posted another question. Thank you, Anne. Building off Alba's questions, given that some insect vectors, i.e. meadow spittlebugs, harbor lower cell counts than the blue-green sharpshooter, that's a great insect name, a few thousand versus tens of thousands, would combining this technique with digital droplet PCR provide a good means of measuring the growth rates or phases within the insect vectors?
And yes, it could totally be applied to that. You know, like the number of cells that an insect species can harbor and the growth rates. I mean, it doesn't mean that because the spittle-bugs harbor less cells that the growth rates is lower. But yes, this could be done to determine it.
Interesting. Okay, I think this has been great. Anne, thank you, and thanks for answering all these questions. We've had a terrific set of questions and ideas about applying this technique and technical advance. Where would you like to see this work go? What would be, you know, where do you think it would be most interesting to apply it?
Well, I think there are lots of possibilities to apply it. Yes, looking at whether, I mean, testing different treatments on Xylella growth rate and see if it can actually like impede the growth rate would be actually pretty cool.
Yeah, that would be very cool.
There's one more question.
Okay, I think we'll take the last question and then we will stop. So this last one is Juan Carlos Diaz-Vici. And he asks, do you think that this technique would be useful to evaluate root colonization by rhizospheric bacteria?
I think it's worth trying it.
Yeah, I think that would be very cool too. Okay, well, thanks so much, Anne, for the very interesting seminar, and for everyone for your questions and ideas. Just to remind you that the details of all of this, if you have more things that you want to check back on or wondering how she does some of these things, the details are all in the paper. So I hope that you'll go back and take a look at the article. Ashley has pasted it in the chat, although I think it was kind of near the top. She has reposted it. So there you have it. Go find it, go take a look. And remember that if you have questions that come up later, you can always email me and I will forward them on to Anne. Or also remember that every paper comes with a corresponding author with their email address. And it's for exactly such things like this, for you to engage with the authors who would like nothing more than to see people use their technical advance. Okay, well, I think we're going to stop there. Thank you, Anne. And we'll see the rest of you hopefully back on December 1st.